Okay, so we're basically ready to start with characteristics, but I wanted to do an example or two of agonist and antagonist before we start there. So I'm looking at the last page in the muscle chart, the last two pages. I want you to focus on the gastrocnemius, the soleus, and the tibialis anterior and tibialis posterior. I want you to think about standing on your toes, which would be extending your foot at the ankle. So out of those four muscles I just named, which of those extend the foot at the ankle? And you can look at the chart or you can do it from memory, depending on where you are. So which ones extend the foot at the ankle? Gastrocnemius. Okay, so we're defining the action as extending the foot, and it's implied to mean at the ankle. So we have the gastrocnemius. Somebody else, are there any others that extend the foot at the ankle? The soleus? And any others, or is that it? Tibialis posterior. Okay, so those are the three that you use when you stand on your toes or you extend your foot at the ankle. So if the action is to extend the foot at the ankle, what would the antagonist action be? Flex the foot. Okay, so antagonist action, flex the foot. And again, it's implied at the ankle. So antagonist muscles would be any muscles that flex the foot. We've got that group of four that we're focusing on. Do any of those flex the foot? Tibialis anterior. Okay, so we have said, or you have said, for the most part, that because the tibialis posterior extends the foot, and the soleus extends the foot, and I can't reach up there, but the gastrocnemius extends the foot. They are agonists with respect to extension of the foot. They work together synergistically with respect to extension of the foot. One of them is the prime mover, meaning it's the strongest. And I'll tell you, in case you want to know, it's the soleus, but you don't have to figure out the prime mover for any movement. I just want you to know what prime mover means. And so the antagonist muscle is the one that flexes the foot. So now focusing on the two at the bottom to finish this up. You have told me that the tibialis anterior and tibialis posterior are an antagonistic muscle pair because one extends the foot and one flexes the foot. Are they also an agonistic muscle pair? Look at the actions. They both invert the foot. So if we're talking about inversion versus eversion, they're an agonistic pair. So now the question is, are the tibialis anterior and tibialis posterior an agonistic pair or an antagonistic pair? Talk. Huh? Absolutely. Are the tibialis anterior and tibialis posterior an agonistic pair or an antagonistic pair? Okay, let's do it this way. Are they an agonistic pair? Why? Yeah, correct, but why? They both invert the foot. Are they an antagonistic pair? Why? One flexes and one extends. So now back to the original question. Are they an agonistic pair or an antagonistic pair? Both. Both. It depends on the action. So see, we started by defining an action. And we came up with them being an antagonistic pair. But what if the action we defined was inversion? Then what would we have come up with? That they're an agonistic pair because they both invert. You want to be able to play this game without looking at the chart. I don't mind if you have the chart tattooed on the back side of your eyelid. I won't check for that. So I won't even know. Sounds kind of painful and take an awfully small font. Let's look at one more example. Then we'll leave this. Looking for the trapezius. Like that's on page 1. Last muscle on page 1. So if somebody said the trapezius is its own antagonist, that's actually true. Why? What do you see? It elevates and depresses the scapula. So you have the ability to contract the superior part of your trapezius and elevate the scapula, or the inferior part of the trapezius and depress your scapula. I know I already said one more, but one more. Looking for the deltoid. There it is. The deltoid is also its own antagonist. Why? It flexes the arm and extends the arm. So now as we move into today's discussion, which is characteristics, we'll see the precision involved in some actions. You know, we'll be thinking about either an Olympic diver or an Olympic figure skater or an Olympic gymnast, or any of you snow skiing or snowboarding. So the word characteristics is misleading because it's a word, but it will occupy most of the lecture this afternoon. Muscle tissue, and I mean all 4 types, skeletal, smooth, all 3 types, so 4 types, skeletal, smooth, and cardiac. They have 4 characteristics. Now listen, let me tell you right up front, this lecture's 90% physiology, and you'll probably enjoy it. It's kind of cool, and you'll definitely prefer it over memorizing the muscle chart. But just because I'm recording, and let me make sure I am. Yep, I have been for 9 minutes and 38 seconds. Just because I'm recording, don't sit there and not take notes. Remember, when you write it down, when you take notes, even if they're illegible, it helps burn it into your brain. 4 characteristics. Number 1, irritability. Number 2, contractility. 1 and 2, irritability and contractility. So that means muscles are capable of being irritated. Translation, muscles are capable of being stimulated. And then characteristic number 2, contractility. That's how muscles respond when they're stimulated. So the technical way to say it is muscles possess the characteristics of irritability and contractility. Explain that to a child. Muscles are capable of being stimulated and they respond to the stimulus by contracting. Now, what system of the body or what structure of the body, there are a bunch of ways you could answer this question. What stimulates muscles? Nerves. So whether you say nerve, neuron, the nervous system, at this point, it doesn't matter. They're all good answers. The point is, muscles don't just have a mind of their own. Oh, the sun came up, I think I'll contract. They contract because the nervous system stimulates them. They're capable of being stimulated or irritated, and they respond to the stimulation by contracting. Characteristics number 3 and number 4 are more important next semester when we're talking about the heart. But that's not to say they're not important right now, but they're more important next semester when we talk about cardiac muscle tissue. So number 3 and number 4 are extensibility and elasticity. Just like you might remember reading in some of my documents in histology with elastic connective tissue. Extensibility means it can stretch like a rubber band. You can stretch a rubber band. And a rubber band returns to its original length because it is elastic. As little kids, we take those properties to be, one and the same. No, chances are, unless you remember it from your 7th grade life science class, nobody ever tells you that a rubber band being stretched and recoiling are actually 2 totally different properties. Extensible, it stretches; elastic, it recoils. Now remind me, what did you say stimulates a muscle so that it contracts? A nerve or the nervous system. Okay. So we're going to look at my elbow again. Biceps brachii. So a motor nerve stimulates the muscle and it contracts. And because we're talking about the biceps brachii, when it contracts, we would get flexion at the elbow. The motor nerve zaps the muscle. Zap! It responds by contracting. It all happens because the muscle is capable of being irritated and capable of contracting in response to the irritation. Motor nerve. That's as opposed to sensory nerve. Some of you will recognize some of this from a Psych 101 class, and if you don't, that's okay. But motor nerves run, or rather motor nerves leave the central nervous system. Sensory nerves travel to the central nervous system. Why'd you guys write slow? Told you to take. I want you to touch something. Did you feel it? You could tell me it was smooth or rough, yes, couldn't you? Was it hot? Was it cold? Was it painful? You could answer all of those questions, correct? So when you touch the thing, whatever you touch, okay, I'm going to touch this. Receptors in my integument are activated and sensory pathways conduct the signal from my fingertips, through my fingers, through my hand, through my arm, to my shoulder, my axillary region, and then to my spinal cord, and then up to my brain. And I perceive that texture with my parietal lobe. Parietal lobe, parietal bone, same location. So sensory nerves are conducting signals from somewhere, the toes, the fingers, the lips, to the parietal lobe, that specific part of the central nervous system, and you feel whatever it is. If I make the conscious decision to grab my water bottle, the muscles that I use to reach out and grab it, bring it to my face, unscrew it, take it, etc. All of those signals originated in the frontal lobe. And it sent the signals from my frontal lobe down my spinal cord, out to my arm, innervating the muscles that were necessary to do that action. So motor, think frontal. For sensory, think parietal. Now these signals travel at lightning speed. Go ahead, touch something again. How long did it take before you felt it? It was instantaneous, wasn't it? No, it wasn't. It appeared to be instantaneous because it's traveling so fast. Let's say that the distance from my fingertips through my arm to my cord to my brain, let's call that 3 feet. Well, how long do you think it takes to travel 3 feet if you're traveling 100 meters per second? That's essentially a football field in 1 second. You're like me, you'd rather have American units. 237 miles an hour. So if you're traveling 237 miles an hour, how long does it take you to travel 3 feet? I know, let's go out on powers and find out. We'll put 2 marks. Mark A, 3 feet away, mark B. How long, and here you come at 237 miles an hour. And how long does it take you to go 3 feet? Instantaneous, but it's not, but it appears to be. These signals are action potentials. That's the name of the signals. They are an electrochemical signal. similar to electricity. And you should think of them as electricity until you come to really understand them. Let's fast forward in the notes, very last page. Look what we talk about. Look at that. Action potentials. So we'll spend 30 or 45 minutes talking about action potentials. For now, think of them as electricity. So an electrical-like signal leaves your brain and moves out into the muscles in your leg and you kick the ball. Or an electrical-like signal leaves your brain, your frontal lobe, and moves out into the muscles in your arm and you pick up the box. You touch something, you say, God, that's sharp, that's hot, whatever. An electrical-like signal traveled from your hand through your arm up to your parietal lobe. And these signals are traveling 237 miles an hour. Similar to electricity. Electricity is the flow of electrons. You know how water flows through a pipe? The pipe under your sink, or the pipe that feeds your shower, or the pipe that feeds the garden hose, or the pipe that feeds an ice maker. Water flows through pipes. Well, electrons flow through wires. And that's what electricity is. Literally. The bigger the pipe, the more water can flow through it. The bigger the wire, the more current can flow through it. Well, guess what action potentials are? Because remember, they're similar to electricity. Action potentials are the flow of sodium ions. What does a sodium ion have in common with an electron? They are both what? Charged. They're both charged things. They're both charged entities. That's what makes action potential similar to electricity. Electricity, electrons are flowing through a wire. Action potentials, sodium is flowing through a nerve cell. Again, we will come to really understand action potentials, but it won't be today. It won't be next week. All right, so here's where we are. Four fingers this time instead of three. Oh, good God. Muscles can be stimulated or irritated by motor nerves, and they respond by contracting. But if it's that simple, if that's all it is, then it's on or off, yes or no. It's binary. But it's not binary. Let's continue with the biceps brachii. What if the biceps brachii contracts with enough force to curl this paperclip? And now I pick up my phone. And it has to contract with enough force to curl my phone. And now I pick up the fictitious bowling ball. And it has to contract with enough force to curl the bowling ball. So it's not contract or not. It's not on or off. In other words, these lights are binary. They're on or off. There's no dimmer switch over here. You know what a dimmer switch is. There's not one of those. The lights are on or off. Well, we know that with your muscles, it's not on or off, because muscles have the ability to contract with varying degrees of force. What if an Olympic figure skater wants to do a triple axel? Just in case you don't know, my God, the men now have to do a quadruple axel, and they're required to. This kid the other day did something like 5 quadruple axels and won the world championship. How many did he do?
My gosh, I want to say 4, or was it 5?
It was 4 or 5. It was unheard of. It was just, I mean, he might as well have flown like a bird. But imagine if you hurl yourself into the air to do a triple axel. What if there's too much force? You'll over rotate. Not enough force. You'll under rotate. If I go to pick up this, I lost my paperclip. If I go to pick up this paperclip and I use the same amount of force I would use to pick up the phone, I'd hit myself in the head. If I go to pick up the phone and I use the amount of force I would use to pick up the paperclip, I wouldn't be able to lift it. So it can't be on or off. There's got to be a way to vary the amount of force that a muscle exerts. And not just a muscle. Think about that Olympic figure skater. And if you don't like that, think about the Olympic gymnast, the Olympic diver, and whatever you want to picture them doing. How many muscles in their body contract? And they all have to contract in the right order, don't they? And when each muscle contracts, not only does each muscle have to contract in the right order, but the right part of the muscle has to contract. Because as we just saw, muscles like the deltoid, the trapezius, they're their own antagonist. If you need to elevate the shoulders and you innervate the inferior trapezius, you'll depress the shoulders. So the right muscles have to contract, the right part of the right muscles in the right order, and each one with the right degree of force, as that Olympic athlete does their bleh to win the gold medal. So, how do we break it down so that it's not binary? It's not on or off. In other words, instead of having that light switch over there, we need to install a dimmer switch. So a muscle is divided into many muscle cells or muscle fibers. Same thing, interchangeable. And a nerve is divided into many nerve cells or neurons. So if we look at a cross section through this nerve or a cross section through this muscle, we would see many individual nerve cells, or if it was a muscle, many individual muscle cells. You can fill that in the rest of the way if you want to. So if you're going to make the biceps brachii contract to flex the elbow, you have the ability to send signals through any number of these individual nerve cells. You can send a signal through 10 of them, 100 of them, 10,087, 387,042, and your brain knows. How many of these individual, and again, there's millions of these nerve cells in this nerve, not whatever I've drawn, 22. Millions of them. And your brain knows how many of those individual nerve cells to send a signal through, depending on whether you're curling a marker or curling a phone, or you're at the gym curling 5 pounds, 10 pounds, 20 pounds. A single nerve cell innervates a small group of muscle cells. So the nerve and the muscle are divided into millions of individual cells, and one nerve cell will innervate or stimulate a small group of muscle cells. This combination is called a motor unit. So when it comes to how much force does my muscle need to deliver, it comes down to how many motor units are we using? 156, 10,087, 387,052, and your frickin' brain, your frontal lobe, knows how many of those motor units to use. It's learned how many, because what happens the first time you try something new? Let's say you're a skier, but you've never gone off a jump. What might happen the first time you go off a jump? You may crash. Or first time a little kid tries to dive into the swimming pool. They may crash. I don't know if anyone in the room is a figure skater or not, but let's say none of us are. So what would happen if we went to a skating rink and put on skates and hurled ourselves into the air and tried to do just a little double act? We die. Okay. So your brain learns. You're not just born knowing this, but your brain learns how many motor units it takes for all of these different things. All right, crazy stuff. Now, if you hate this, well, throw it out and I add 100 muscles to the muscle chart. The choice is yours. I won't tell you how the morning class voted. So what do you want to do? Keep the physiology and keep the muscle chart the way it is, or dump the physiology and memorize more muscles? What do you want to do? Okay. I knew that's what you would say. It's because we're intelligent creatures. I've said so many times, memorizing anything is simple. It's just boring. My dad, he was a trip when it came to memory, at least short term. You could take a deck of cards, a shuffled deck of cards, and just lay them, how many cards are in a deck? 50, 52, and just lay them all out there, and then pick them up, and you could say, what was the 32nd card? What was the third card? What was the 11th card? And he could tell you. That's a trip. Okay. Next topic. This is topic 9, Anatomy of a muscle. So we'll be starting at the gross level. We can see it with the naked eye. We'll be going down to magnified thousands of times. We're going, imagine starting with something and subdividing it. What if we subdivide the world? Well, we start with 9 continents. What continent do we live in? North America. What are the subdivisions of the North American continent? Well, the United States is one. That's where we live. Does the United States have subdivisions? They're called states. Do the states have subdivisions? They're called counties. Do the counties have subdivisions? They're called cities. Do the cities have, yes, neighborhoods. That's what we're going to do with a muscle. This is super cool. Okay, so we'll be using this picture. Probably be times you want to take a picture of it. So we start by peeling the skin off of the subject's arm, and we're looking at the extensor muscles. And we're going to take one of those muscles and we're going to follow the arrow and bring that muscle down here. And we see that that muscle has actually 2 outer boundaries. One of them is not in the picture, so I will add it. So it has 2 outer boundaries, the fascia and the epimysium. The fascia and the epimysium surround, bind, and hold together the subunits of a muscle. Yeah, what? The fascia and the epimysium surround, bind, and hold together the subunits of a muscle. If these markers are the subunits, my hand is holding them together, isn't it? My hand is surrounding, binding, holding together the markers. So my hand represents the fascia and the epimysium, and it is holding together those subunits. The fascia and the epimysium together surround, bind, and hold together the subunits of a muscle. This is at the gross level. Remember, we just skinned our subject and we removed one muscle from the antebrachium and we laid it on the table. We're looking at it. There's a superficial layer, fascia, and just deep to the fascia is the epimysium. Together, they hold together the subunits. I have not named the subunits yet, so you're not missing anything. The fascia and the epimysium do something else. Baxter, yell at me if I don't go back to the main board when I'm through over here. The fascia and the epimysium do something else. They form the tendon. So you learned in histology that tendons connect muscles to bones, but nobody ever asked me how. Are the tendons stapled to the bone? No, they're not. Are they duct taped? No, they're not. It works like this. So there's a muscle, there's a bone. There's the tendon connecting the muscle to the bone. This outer boundary of the muscle is the fascia. Now go back in time, please, to the anatomy of a long bone. What's that covering? What's that outer covering of a bone, any bone? We were talking about long bones, but it covers any bone. Periosteum. So that's what this is over here surrounding the bone. So look, guys, the fascia of the muscle is continuous through the tendon with the periosteum of the bone. That's how tendons and bones are connected, not with staples. Now, use the picture on the main board if you have to, but what's just deep to the fascia? The epimysium. So the epimysium surrounds the muscle, extends out into, contributes to the structure of the tendon, but it does not surround the bone. So recapping. Main board. The fascia and the epimysium together surround the entire muscle. They bind and hold together the subunits of the muscle. The fascia and the epimysium also, small board, form the tendon. I hurt so bad, I almost wish one of you just shoot me. But I mean, make sure it kills me, don't let me just shoot me in the leg.
Shoot you the kneecap. There you go.
Thank you.
Take the pain away from your hips though.
Yeah, well, that's kind of like when every time I get electrocuted, it takes the pain away. All right. So, repeating myself to set up what I'm going to say next. The fascia and the epimysium surround, bind, and hold together the subunits of the muscle. The subunits are these things. And what we're going to do is we're going to grab one of those and pull it out. And we're going to follow the arrow, blow it up. So this thing over here is one of these. So those subunits are called fascicles. Fascicles. So now when we say the fascia and the epimysium surround, bind, and hold together the subunits of a muscle, the subunits are what? Fascicles. Now take a look at that fascicle. Does it have an outer boundary? It's called the perimysium. So what is the, or what are the outer boundary or outer boundaries of a muscle? Epimysium and the fascia. Which is deep, which is superficial. Epimysium's deep, fascia is superficial. And what are the subunits they're holding together? Fascicles. And each fascicle has an outer boundary. What's it called? The perimysium. So a minute ago, my hand represented the fascia and the epimysium holding together the subunits of the muscle, which are fascicles. Now my hand represents the perimysium holding together the subunits of a fascicle. The subunits of a fascicle are those things. Okay, like I said, we're subdividing. So the perimysium surrounds, binds, holds together the subunits of a fascicle, which are muscle cells, also called muscle fibers. But I think I've already told you that, right? In the little chart when I broke down a nerve and a muscle cell, muscle fiber, interchangeable. How many outer boundaries does a muscle have? 2, fascia and epimysium. Based on what I've told you, and I've told you the truth, how many outer boundaries does a fascicle have? 1. Now I'm making that point for a reason, because we're going to have something else in a minute, and it's not going to be 1. So the outer boundary of the fascicle is the perimysium, and it holds together the subunits of a fascicle, which are cells. So each of these is a muscle cell, and there's something in those muscle cells that I hope you recognize from Monday. Look at the nuclei. They are peripherally located. We didn't look at this picture on Monday, but we looked at this one. And we talked about the fact that skeletal muscle cells are huge, multinucleated, cylindrical, and have peripherally located nuclei. When we see the nuclei there, we can tell they're peripherally located. But when we cut the muscle in cross-section, all we see are things like we see right here. Peripherally located nuclei. So that's what all of those green spots are. And you don't see one in the middle, like, you know, a typical cell. First time in your life you ever talked about cells, whenever that was. They always draw the nucleus in the middle. So the perimysium holds together the subunits of a fascicle, which are muscle cells. Looking at one of those muscle cells, do you see subunits? You do. So we better blow up a cell. So let's take one of these cells and bring it down here and blow it up. So there's a single muscle cell. And again, we see the peripherally located nuclei. Muscle cells have an outer boundary called the endomysium. Now, these words are similar. What was not the superficial most, but just deep to the fascia? What's the outer boundary of the whole muscle? Epimysium. And the outer boundary of a fascicle, perimysium. And the outer boundary of a muscle cell is endomysium. Now, I don't know when or where you guys took biology, but when I took biology in middle school, high school, and college, last semester I took Biology 111, I learned that the outer boundary of any animal cell was a cell membrane. The outer boundary of this muscle cell is the endomysium. How many outer boundaries does the whole muscle have? 2, fascia and epimysium. How many outer boundaries does a fascicle have? 1, the perimysium. Well, muscle cells have 2 outer boundaries. So 2, 1, 2. 2 outer boundaries of a muscle cell. I can't reach endomysium, but they are the endomysium and the sarcolemma. The sarcolemma is just the new name for the cell membrane. Now, the following questions will not be in order, so think before you answer. What are the subunits of a fascicle? Muscle cells or fibers. What are the subunits of a whole muscle? Muscle fascicles. So the subunits of the whole muscle are fascicles. The subunits of each fascicle are the cells or fibers. As you look at the one cell, does it look like it has subunits? So just as the fascia and the epimysium surround, bind, and hold together the subunits of the muscle, which are fascicles, and the perimysium surrounds, binds, and holds together the subunits of the fascicle, which are cells, the endomysium and the sarcolemma surround, bind, and hold together the subunits of a cell, which are myofibrils. So how many outer boundaries does a fascicle have? And how many outer boundaries does a cell have? And how many outer boundaries does the whole muscle have? And what are the outer boundaries of a cell? The outermost. Endomysium and just deep to the endomysium. Sarcolemma. And it probably won't be today, but we'll come to understand where that prefix sarco is coming from. Do you see any other part of the cell that's been renamed with that same prefix? And what do you suppose the sarcoplasm is? Cytoplasm? There's another one they renamed. I don't know why they didn't put it in the picture, but they renamed the endoplasmic reticulum. What do you suppose they called it? The sarcoplasmic reticulum. So there's no longer an ER, there is an SR. Again, we will come to understand where all this sarco crap comes from. There's a reason for it, and I don't think we'll understand it today. The morning class didn't understand it today. Well, we didn't get to it for the morning class. So I doubt we will this afternoon. I don't want to. Redundant here, but what are the subunits of a muscle cell? Myofibrils. Okay, I'm blowing that cell up. I don't see any subunits of the myofibrils, do you? So does that mean there aren't any, or just that we need more magnification? We need more magnification. Like the myofibrils are the city. So we're almost all the way. The myofibrils are the city, but now we've got to divide the city up into neighborhoods. So a myofibril is divided into myofilaments. That's almost cute. You know, you can almost just see God and Darwin sitting at a table at Village Inn. And they got this far. And they said, should we stop here? And one of them said, as they lit their pipe, no, it'd be too easy. We have to subdivide the myofibrils. And then took a drink of the coffee. Can't you just see that? All right, there are two myofilaments. Actin and myosin. And some of you probably heard of those proteins. You may know them as the contractile proteins of muscles. Actin is thin relative to myosin, which is thick. And what we need is a different picture. More magnification. I mean, this gives us more magnification, but we don't have the resolution to see subdivisions of the myofibrils. Now, first of all, I want you to tell me, are we looking at a muscle, or are we looking at a fascicle, or are we looking at a cell? And how do you know it's a cell? Because the sarcolemma is labeled. And we know the sarcolemma is the outer boundary, well, not the outermost boundary, but we know the sarcolemma is one of the outer boundaries of a cell. If this was a fascicle, then the label that says sarcolemma would instead say what? Perimysium. And if this was the whole muscle, the label that says sarcolemma would say epimysium. Okay. So we're looking at a cell, and so just quickly to not lose perspective, in this picture, these things were the myofibrils, but we've blown it up now to the point where these are the myofibrils. Now we can see the subunits of the myofibrils. And when we grab this one myofibril and we pull it out, we can again see the subunits in the cross-sectional piece here on the end, but we can also see them longitudinally. Now the thin one, as I've already told you, is actin, and the thick one is myosin. And I agree with you. It's a cheesy picture because I don't see any difference in the thickness of actin versus myosin. Okay, so they could have done a better job with their picture. You'll be happy to know that the myofilaments don't have subunits, at least not in cross section. In longitudinal section, they do. Now, do you remember when this was Monday, when we were talking about skeletal muscle tissue? And you can, look in your notes from Monday if you want to, but, it's voluntary. We associate it with movement, posture support, but also things like childbirth, speech, defecation, smiling, our facial expression, speech. So a lot more than just movement. And then histologically, we said the cells were big cells. They're cylindrical, multinucleated, and they are, what do you see there? A word I hadn't used. Striated. They're striated. And I said those striations were an illusion, didn't I? They're not really there. Okay, so this is a cell. I'm looking at the bottom just because it's the one that's labeled. But this is a cell because I see the sarcolemma labeled. And within the cell, I see myofibrils labeled. Do what, Leda?
I was saying that there's
Yeah, exactly. Yeah, we see the subunits are the myofibrils. And this is just a much larger view of a piece of the muscle cell. One more time, we've got those striations. They're not really there. So why do we see them? Well, notice that we have an area like this where we have just the thin filaments. And we have an area like this where we have just the thick filaments. And then we have this area right there where the thin and the thick overlap. Those striations that we see with lower magnification are right here. Here's why they're right there. We use dyes to stain tissue. Now, the words dye and stain are misused all the time. In microbiology, someone will say, "Will you hand me the stain?" No, I can't hand you the stain. I can hand you the dye and you can use it to stain the tissue. They're like affect and effect, or take and bring. My daughter said when she was a baby, "Dad, can I bring this to Nana's?" No, you can take it to Nana's and then you can bring it home. Can't bring it to a place where you're not. Where'd you go to? She said, "Well, Dad, I'm only 2." I said, "Okay, I'll give you a little grace. No punishment this time." But the stuff in the bottle is a dye. You use it to stain a tissue. Different dyes have affinities for different types of molecules. So you can have a dye with an affinity for things that are acidic, or a dye with an affinity for things that are basic, or a dye with an affinity for protein. And that's what we've got right here. Because these myofilaments, I erased it, but these are proteins. So when we pour a dye on this tissue, the dye binds the protein and stains the protein. The more protein there is, the more of the dye binds it, and the darker it stains it. So in this area, where we have just actin, and it's the thin one, that's going to be the lightest area. In this area where we have actin and myosin overlapping, there's a lot of protein for that dye to bind. So this is going to stain the darkest. In this area here, it's just one of the filaments, but it's the thick one. So this will be darker than this, but this will be lighter than this. So we get these patterns because of the amount of protein that there is for the dye to bind. So when we stain muscle tissue, we get super light areas, super dark areas, and things in between. And we'll talk about this picture officially, formally on Monday. And that's why I don't even want you looking at the labels right now. I just want you to appreciate that the dark areas are where there's more protein for the dye to bind and stain. And so there's more protein here than anywhere else. So more dye binds that area, so that area stains darker. So when you don't have this much magnification, great, big, bigger than life, then that area where actin and myosin overlap goes up like this. Those vertical stripes, they're not really there. That's just where actin and myosin overlap. This is wild, isn't it? I'll ask you again. You want to continue with muscles and how they work? Leave the muscle chart alone, or if you hate this, we can dump all this and memorize more muscles. I don't know about you, but I hate sitting in the corner staring at that muscle chart. Horrible. So we'll leave the muscle chart alone and we'll continue with physiology. The part of the class that we started today is, by the way, the most interesting part of the class because we are complex, intelligent creatures. Functionality, physiology is far more interesting than anatomy. And generally speaking, functionality doesn't even have to be memorized, it just has to be understood. Functionality is more like watching a movie and telling your friend about it. So I watched The Accountant last night. That's a pretty good movie if you haven't seen it. It's on Netflix. And I'm super tired because of all this. I actually fell asleep and had to rewatch the last 30 minutes. But that's not because it was a bad movie. It was just because of where I was physically. But I could tell you about the movie. And I didn't take notes when I watched it. I didn't memorize any part of it. I didn't repeat any part of it over and over. I just watched it. That's the way physiology is. You just understand it. You don't have to memorize it. I know you do have to memorize it at first. And then the further you go with it, like those of you that take pathophysiology, everything will just make sense. And don't dare take pathophysiology before you take AMP2. It's allowed, but it's a huge mistake. It's absolutely stupid. Do not do it. Oh, I was about to erase those lines. Good Lord, Larry. Okay, so we're going to pick up. We're still in this anatomy of a muscle, gross down to the bands. But we're going to pick up with what are the names of the bands and where's all this sarco terminology come from? That prefix, we see sarcomere, sarcoplasm, all of that. Like I told the morning class, while I talked about class even started, the very first email and or the alpha document, the importance of keeping up extremely. When you come through that door on Monday, please, 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 it needs to be like we just took a 5-minute potty break right now and we came right back. It can't be like we were gone for the weekend. And you come in here and you don't know what you've got to understand everything I just talked about. So you've got the notes you just took, got the recording, got a textbook, maybe. If you don't, you've got 1,000 textbooks online. So please come in on Monday, understanding, being right where we are right now, okay? But now, hold on, Raquel. But now, what else do you have to do this weekend? Muscle chart, rote memory. And what else do you have to do this weekend? Prepare for the cadaver. You got to study, a picture of the gross anatomy of the anterior musculature so that when I show you the muscles next week, when I say this is the vastus medialis, this is the vastus lateralis, this is the gracilis, this is the pectineus, and you're hearing perhaps a lot of words that you've never heard, the cadaver looks just like the picture, and it all makes sense. Raquel.
There's a great video I found on YouTube last night that kind of covers a lot of stuff that we're talking about right now in class. It's on Crash Course, Muscles 1 and 2. It helped me a lot to understand what we're talking about with the physiology part.
Thank you. And speaking of that, and this is not YouTube, but it reminded me of something that I wanted to tell you. So I'm going to D2L, going to the online resource module. Okay, so down to the online resource module. Remember that the online resource module has 2 sub-modules. I'm going to open the video sub-module. Am I in AMP2? Idiot. Online resource module. 2 sub-modules. I'm going to open the video sub-module. Look at those videos that have the word muscle in their name. Now you're not ready for those this weekend. I mean, it doesn't mean you can't go ahead and watch them. But after Monday's lecture, they'll make a lot more sense than they make today. Now, this first one that doesn't have the word muscle in it, but you can see it's called motor units. Recognize that? Recruitment. That one you're ready for. The other ones you're not. Now, those videos, you're going to see the exact same videos somewhere else. So again, I'm going to the online resource module. This time I'm going to open study resources instead of videos. And in study resources, I'm going to go to muscles. And you see those same videos. I'm pointing this out just so you don't think, oh, there's these 5 here and there's these 5 somewhere else. I just put them in 2 different places. Now, as far as watching these videos, it's going to depend on your system. First of all, whether it's a PC versus a Mac. Some of you will be able to just click the video and watch it in D2L. Some of you will have to download the video and save it to your device. But you're all intelligent people. You grew up with computers, so I'm sure you can figure it out. And on the Mac, they play under QuickTime, I think. On the PC, they play a Windows Media Viewer or something. But sometimes you have to download the app if you don't already have it on your device. But I mean, again, you're intelligent creatures. You can figure that out. All right, so let me end the recording. Oh, darn, the recording we haven't been recording. I'm kidding. Wanted to give you a quick heart attack. All right, record.